It's a pleasure to introduce a fellow Floridian, Michalina Janiszewska. She grew up in Wrocław, which is one of my favorite little cities in Europe. She went to the university there, went on to do doctoral work at Lausanne, and then postdoc work with Nelly Polyak at Harvard. She has published extensively at very high-level journals, very interesting articles, and you know, it's really it's a pleasure to hear about her work. So, Michalina, she's going to speak on genetic heterogeneity and glioblastoma.
All right, thank you so much for the kind introduction and for the invitation to speak today. It's really great to meet new people in this online reality that we are still partially living in. So I'm going to talk to you today about an ongoing study that we started in the lab that focuses on in situ analysis of single cell genetic heterogeneity in glioblastoma. So just to give you a little bit of a background on glioblastoma, this is the most frequent malignant brain tumor. And despite the fact that relatively its incidence is not very high because it's about 3 cases per 100,000 people, it's associated with a really excessively high morbidity and mortality. There's only about 7% of people who are surviving beyond 5 years, which is really a terrible outcome for those patients, and there is no targeted therapy that has been developed for those tumors. So the lack of effective treatment for GBM is actually associated with its extreme heterogeneity. So the studies geared towards profiling of these tumors shown that basically you can classify each GBM into the distinct phenotypes based on RNA expression. So you can have a classical proneural and mesenchymal type of tumor, but then actually with the newer technologies that enabled single cell sequencing, we learned that every single tumor seems to be kind of a mix of those different subtypes. And the most recent studies actually identified that there's probably 4 major subpopulations of cells within each GBM tumor that can be classified based on their gene expression to sort of resemble the OPC-like cells, NPC-like cells, AC-like, or mesenchymal-like. And basically what I wanted you to take away from this slide is that each one of those sort of specific phenotypes is actually associated with certain genetic features. So basically amplification of PDGFRA, CDK4, and EGFR are the features that are very frequently present in distinct subpopulations of cells within these tumors. And this was this sort of genetic variation within the GBM tumors has been also observed before we had access to those single cell technologies. So there was a paper in 2011 showing very clearly, just using standard fluorescence in situ hybridization techniques, that amplifications of EGFR, MET, and PDGFRA actually occur in distinct subpopulations of cells within the same tumors. So we wanted to sort of not only take into account this genetic mosaicism of GBM, but also to learn something more about the tumor microenvironment. Because in every GBM, there is a stark variety of different regions within the tumor that consist of slightly different cell types. And we sort of are, of course, interested in thinking about this as a grand ecosystem where tumor microenvironment diversity could actually be driving selection of different cell types with particular traits in those tumors. So in order to study this interaction between the genetic and microenvironmental diversity, it's very hard to do that in sort of experimental systems, because it's very hard to replicate the complexity of those ecosystems in vitro models or with animal models. So because of that, we actually started this study by profiling 17 GBM cases. So we obtained the full blocks of GBM tumors, and we selected 3 to 4 different cores per each one of those biopsies, and we constructed this tissue microarray. So basically, this tissue microarray contains 68 cores from 17 samples and 3 to 4 cores per tumor. And we did a very extensive profiling on that. And this study was led by Kasper Valentinovich, who is a postdoc in the lab, together with Dalit Engelhardt, who is a postdoc with Franziska Mikor at Dana-Farber. So with this sort of extensive profiling, what we've done was we used different techniques that allowed us to profile 79 protein markers, but also the single cell FISH quantification and single cell third promoter mutation assessment. So I'm going to talk about all of these aspects briefly in a moment. So first of all, we performed on this tissue microarray, we performed a digital spatial profiling. So as I mentioned, we had 79 protein markers that are geared towards tumor biology, neurobiology, and immunology. And this assay is based off the nanostring DSP GeoMX platform. So for those of you who are not familiar with this, it's basically a simultaneous binding of antibodies targeting all these different markers at the same time. And these antibodies have oligonucleotide tags attached to them. So when you select a region of interest that is illuminated with UV light, these oligonucleotide tags get released and they can be quantified. So basically this allows you to quantitatively assess protein expression in particular regions of interest. And for us, we selected 96 regions of interest in our tissue microarray. And this is just showing you more or less how it looks for some of those areas. So you have those areas that look fairly similar to each other, but then in some other cores that we looked at, there was quite a lot of variation between the different cores. So basically we selected the regions based on the staining for CD45, which marks the immune cells, the smooth muscle actin, which marks the blood vessels, the GFAP that should be expressed by the GBM cells and probably also astrocytes and the DAPI. So with this platform, what we were able to do was to look at expression of all of these different proteins at the distinct locations within the tumors and profile all these samples. So first, we just looked at a simple correlation of protein expression just to check whether our data actually makes sense. And indeed, we found many relationships that were expected. So for example, that the phospho-tau proteins actually were co-expressed in the tissues, and we also found, for example, that all the immune markers were actually also sort of expressed in those different regions. And here we also saw that there was an anti-correlation between immune markers and neuronal markers. But of course, we were not really interested in globally assessing the expression, but we wanted to really see what are the differences between sort of the inter-tumor variability, so comparing the different biopsies from different patients versus the intra-tumor variability, which would be really comparing the differences within a particular biopsy. So when we did that comparison across all of our protein markers, what we saw was that there was a increase in the proteins that were sort of correlated with vasculature in those sort of statistics that were associated with the more intra-tumor variability. So that would suggest that the blood vessel markers are really more diversely expressed within each one of our biopsies. And because actually with the assays that we performed, we are not really destroying the tissue and we have all the imaging, we can actually go back to the images and see really whether what we find with this profiling is actually true. So this is just showing you the diversity of vascularization in one particular tumor biopsy, but two different regions. So you can see that there's quite a large difference in the sort of distribution and quantity of those blood vessels in this sample. And this is just to quantify. So basically based on the imaging, we also quantified all of those blood vessels within each one of our cores. And you can see that for every tumor, there's quite a lot of distribution. So there's a lot of variability within a tumor for the vascular density. So that was sort of the summary of one aspect that we profile, but we then went back to the same slides of the tissue microarray, and we performed fluorescence in situ hybridization on those to look for EGFR, CDK4, and PDGFRA that I mentioned before to you, that those are the 3 amplifications that are associated with the distinct subpopulations of GBM cells that are probably present in most of the tumors. And then we also performed another assay that is called Starfish, which allows us to look at the third promoter mutation in the intact intact slides, and that allows us for quantification on the single cell level. I'm not going to go into too much detail on that because that's sort of we're going to be out of time if I go through that, but that's just to mention that we can actually do that. So what we've done with the fish is that this multicolor, 3-color fish allowed us to really count all of the signals in each individual cell and then to classify in each individual cell into a particular genotype. So here we had genotypes that were consisting of combinations of those 3 amplifications that we looked at, CDK4, EGFR, and PDGFRA. And what you can see in this graph is that while there was quite a lot of cells that didn't have any of these amplifications, so we call them either neither or just other amplifications possibly, we saw that basically we had quite a lot of cells with CDK4, EGFR, and combination of EGFR and CDK4 amplifications. So based on this, we actually decided to stratify the tumors based on the co-occurrence of those 3 main subpopulations that we identified. So the cells, the E cells, which have just the amplification of EGFR and a standard copy number of CDK4, the C cells, which have high copy number of CDK4, but normal copy number of EGFR, and then the EC, which have both of those amplifications. And what we found was that basically trying to see what are the odds ratio for co-amplification of EGFR and CDK4 within individual cells in a tumor, we were able to actually classify tumors into sort of those 3 distinct groups. So we have, based on the odds ratio, so we have the odds OR low tumors that basically have low frequency of the cells with the co-amplification. And then we have OR high tumors that have high odds of having the co-amplification present in a single cell. So based on this classification, we also looked at overall frequencies of distinct cell populations within these 3 groups, so OR high, OR low, and OR medium. And what we found was that, as expected, in the E cells, so the cells with the EGFR high copy number were high in the OR low tumors. So basically you can see here the green ones. And what was interesting is what you can see actually on this image already is that in OR low tumors, we did observe quite high copy number of EGFR itself per individual cell. So we decided to actually look at that a little bit closer. And what we found was that indeed this difference was actually significant. So the OR low tumors had a really high copy number of EGFR, but these differences in copy numbers were not present when we looked at CDK4 itself. We also looked into the diversity of the copy number using the Shannon diversity index. And we also saw that the cells harboring the EGFR amplification in the tumors that are sort of associated with the OR low phenotype were really way higher in terms of the diversity. So that's prompted us to think that probably this high copy number and also high variation of the copy number in those tumors could be driven by extra chromosomal DNA. So this would suggest that basically these tumors have a very distinct evolutionary trajectory, and we're doing additional experiments right now to actually look into that and confirm that. So next, because we had all of this information actually was obtained from in situ analysis, we could go back to the locations of each individual cell and then quantify relative distances between the cells of the same genotype, which is shown on the left-hand side. Or we could also do a clustering score, which basically allows us to look at the relative sort of distribution of the cells within the tissue for each genotype, basically how close, tightly together the cells are present within the tissue. So with this type of analysis, we didn't really find anything unexpected, because in the OR low tumors, where there is more cells harboring the EGFR amplification, we found that there is lower distance between E cells, which is kind of expected just because there is more of them. So that was one aspect that we measured, but wasn't super exciting. But then what became very exciting to us was when we started looking at correlations between the expression of different protein markers and our genetically based classification of tumors. So again, we are looking here at the expression, protein expression, with divided by our classification based on the genetics. And what we found is that basically for the oral low tumors, the protein that was significantly differentially expressed was EGFR, which is kind of expected, because as I said before, we saw that there was way more EGFR high copy number cells, while on the side of the OR high tumors, there was quite significantly higher expression of all of the markers associated with the immune response. And what was really interesting was this particular marker CD163, which is specifically staining cells that are immunosuppressive tumor associated macrophages. So that's really interesting, because it has not been shown before that sort of genetic drivers could actually in any way correlate with those immune suppressive cell types. So we went back to our tissues again, and we performed the staining of CD163 on our tissues. And we were really surprised to see that actually those tumors that were sort of the most extreme OR low and OR high were really huge differences in expression of CD163. So basically meaning that the infiltration of immune cells, immunosuppressive immune cells within these tissues is really very striking. And then CD163, why I'm saying that this is really exciting and important is because CD163 was also previously associated with survival in GBM. And when we did the analysis of TCGA data sets that are publicly available, we found that this classification based on expression of CD163 is indeed somewhat correlated with the survival of the patients. So that's definitely sort of opening the new door to using the genetic markers as a proxy of immune infiltration and possibly also survival. So in summary, in this study, what we found is that the OR, sort of the genetic classification, can actually be used as a proxy for immune infiltration. So our high throughput single cell in situ analysis provided us with completely new insight into the genetic mosaicism of GBM. And previously, similar analyses have been done, but usually a much smaller number of cells were quantified versus for us, it's more than 2,000 cells per each individual tumor. And previously, all the analysis based on around 50 to 100 cells were not able to capture the extent of co-amplification of EGFR and CDK4. And then our multi-region analysis revealed that there is really high extent of heterogeneity even within a single biopsy, because we have to remember that all of our samples are really indeed just simple small biopsies. So that points to the fact that in GBM we really should be thinking sort of very broadly and very carefully about the overall heterogeneity of these tumors. So our study is still sort of ongoing. So we definitely have a lot of open questions that were brought by our analysis. So we're very interested in how is the co-occurrence of EGFR and CDK4 amplification altering the interplay between the GBM cells and the immune microenvironment of the tumor. And also, we want to know whether EGFR and CDK4-driven immunosuppressive microenvironment of GBM could be responsive to immunotherapy. So I guess we definitely would be very excited to work more with samples from clinical trials to actually better understand what's the genetic diversity and the state of the tumor microenvironment, to see whether those genetic measurements of tumor heterogeneity, whether they could be associated with predicting of the outcome. So that will be very, very interesting. And then we also want to, yes, that's basically what I said. So that we really want to see whether we could use the single cell based genotyping to stratify patients to treatments. So that would be also very, very interesting to explore, because those FISH techniques that we employed to conduct the study, they are relatively cheap and they can be very easily done in the clinical setting. So with that, I guess I'd like to thank all the people who contributed to the study, especially Kasper Valentinovich and Shreya Yadav, two people in the lab who really were instrumental in this work, and our fantastic collaborators, Franziska Mikor, Dalit Engelhardt, and Simona Krista, who were wrangling the data and helping us put everything together. And Christina Vincentelli, who is the pathologist, neuropathologist at Mount Sinai Medical Center, who was actually providing us with all of those samples. And Oscar Schentermay, who's our great collaborator at the Cleveland Clinic, also on the side of providing the clinical material. And with that, I guess I'd like to hear what you think, and I'll be happy to answer any questions that you may have.
Great, thank you so much, Michalina. That's very exciting work. So we do have plenty of time for discussion, which is great. So feel free to post any questions, perspectives, or comments in the chat. And of course, we do really like to hear from people. So if you have a question and feel comfortable asking it verbally, you can use the virtual raise hand function, which you do by clicking reactions at the bottom of your screen and then raise hand. So first, looks like we have a question from Michael Hofer. Michael, you can go ahead with your question.
That was a wonderful talk. And I like your questions for the future. And my question relates to those. Do you have any idea of associations between the patterns that you observe and tumor characteristics, tumor growth rates, tumor shape, such as spiculatedness, which could lead you perhaps to associations with life expectancy and outcomes of treatments?
Yes, so in this particular study, actually, we didn't have that information because those samples are really old, sort of archival stuff that the hospital was getting rid of. But actually, we are conducting right now another study where we are prospectively collecting samples from multiple regions of the tumor. So basically, that would really allow us to, and those multi-biopsy protocols are actually based off the MRI imaging. So then we actually will have a little bit of a better notion of where within the tumor the sample is coming from. So by doing analysis of those samples, I think we will be able to a little bit more get at that question, because I think that's very important, and especially in GBM where we know that there's definitely important differences between the sort of invasive front versus the necrotic center of the tumor.
So you don't have any imaging to associate with?
Not for this, no,
Right.
no,
Okay.
Unfortunately not.
Okay.
Yeah, because basically in order to be able to construct this TMA, we actually had to use the whole blocks. So usually a hospital, it's not very keen on giving up all of the blocks for a patient. So yeah.
Okay. Thanks.
Great, thank you. Next, we'll go to William Parker. William, you can go ahead.
Thank you. I agree with the first question there. That it's a great talk, wonderful work. My question is a little bit general and relates to the number of cells that you really need to catch in a tumor. So can you give us a general idea? Is there 1,000 different genotypes that you're detecting, and how many of them would typically, what % of the total might escape the immunotherapy in a case? I guess that's variable. And then how about chemotherapy? Is that kind of a similar issue where it's just 1 or 2 rare cells that get by, or is it typically a group of cells, or how does that work?
Mm-hmm. So I guess to address the first part of this question, that's really a great sort of problem, right? Because we're always undersampling the tumor. So we're always taking a simple biopsy from probably one location, and then we only take a fraction of the cells that we're actually looking at. So I guess probably this is still not really known how many are needed to really fully capture this, because right now, even with the single cell RNA sequencing, like typically people are using 2,000 to 4,000 cells from a tumor, right? So that's whether that's representative or not. That's something that we have to still look into. And how is it with the with sort of the escape from therapy? I think that's the same sort of answer. That we still don't exactly know. So we are still hoping that by capturing, I don't know, thousands of cells, we will be able to identify those rare clones that are able to escape. But then, as usual, right? If we are only profiling 2,000 cells, and if we can make a confident call based on maybe, I don't know, 10 or 20 cells, that's still a relatively high fraction that would be required to be present in the overall tumor. So that's why I guess I'm really sort of very interested in techniques that are actually able to identify individual cells within thousands and thousands of cells. So that's something that we are trying to do with the with the other techniques like starfish, where we are basically looking at individual cells within the tissue, and we have quite high sensitivity. But I think, yeah, achieving a sensitivity that is higher than 1,000 cells, let's say, that's very tricky just because of the analytical problems.
Thank you. Thank you very much.
Thank you. Next, we'll go to Bernie Crespi. Bernie, you can go ahead.
Hi, right. Thank you for that. I wanted to try to bring things a little bit towards the evolution side, if I may. I'm fascinated with why this type of cancer is so fatal. I'm just wondering to what degree it's a question of the fact that it's in the brain, to what degree could it be a question of the type of cell that's becoming cancerous? To what degree could it be a function of the extreme levels of variability, genetic variability, and so on that you have in this particular cancer? So if we compare this to all of the other cancers, what stands out in terms of the association with the high fatality rate? And then, given that we have some insights about that, say, for example, it's the really high variability, genetic variability that makes it so fatal, then does that give us clues about possible treatments, different types of treatments, trying to push it over the error threshold or that sort of thing? So I think there's several questions in there, but answer the ones that you like, I guess.
Okay, so I guess that would be also a good question to hear Bob's perspective on that, because he may have a lot of insight into this. But let's see. So the question about why these tumors are so difficult to treat. So I think all the things that you've mentioned as possibilities, I think they all kind of add up to the fact that it's so difficult to treat it. So first of all, patients usually who arrive in the clinic, they are kind of, it's sort of a late stage already, right? Because these tumors, sort of patients arrive in the clinic when they already have symptoms. So usually in the neurological symptoms to occur, actually, you really have to have quite severe disease already. So there is no early detection for that tumor. So I think that may be one thing that's probably contributes to the fact that whatever cancerous tumor was initiating the whole process was able to actually gain a lot of time, and sort of the whole population was diversifying over time quite a lot. Another problem, I think, with these tumors is that the cells seem to have quite a lot of plasticity, in the sense that there were several studies showing that at the single cell level, even if you find certain cells to be sort of more differentiated into a particular subpopulation, let's say, a particular subtype of cell, they still can redifferentiate back and forth. So basically, there's quite a lot of probability that if you put a pressure of treatment on these tumor cells, that they will be able to actually adapt and evade the treatment. Another issue with GBM is that there is the notion of the cancer stem cell, which actually is not only getting at the plasticity, but this cell may also be much more resistant to radiotherapy and chemo. So because they don't, there's definitely some populations of cells that don't cycle at the same rate. So there's differences in proliferation as well as other features of the cells. So I think that also contributes to challenges in treatment. In terms of why there is no and another really big problem for GBM patients is that these tumors are not very well sort of defined, in a sense that they very early are highly infiltrated into the tissue. So the current standard of treatment is combination of radiation, chemo, and surgery. And with surgery, basically, the removal of the tumor is never complete because there's already quite a significant dissemination of the cells within the brain parenchyma, and very often, basically, that results in recurrence afterwards. So I think that's probably one of the very important reasons as well why that is. And in terms of how could we, so I think we still don't really know enough about what's actually sustaining the heterogeneity of these tumors and how the whole ecosystem plays in together to be able to target it. Because any therapy that has been tried before as a sort of targeted approach is always ineffective because of all the other potential subpopulations that can actually escape this the treatment. So I think there is still a lot that we have to learn about the drivers of heterogeneity and the ecosystem as a whole to be able to fight this tumor better.
Just a quick thank you for that. Just a quick follow-up. Are there any phylogenies of these of the GBM tumors, and if so, do they have a sort of a shape that differs from, say, other tumors that are less fatal? Can one use the phylogenetics in addition to what you're doing to try to get a handle on how and why it's evolving the way that it is?
So I think there were at least a couple of studies that were getting at that, but I don't think that we have a very comprehensive view of the phylogenesis of these tumors. So I think that's also something that we are interested in the follow-up.
Thank you.
Great, thanks so much for that question, Bernie, and for the response, Michalina. I'm curious, Bob, do you have any additional perspectives on that question, since Michalina was asking if you had any thoughts there?
You're muted.
I agree with all of the difficulties associated with tumors in the brain. There's a couple of interesting factoids. One is that there are circulating tumor cells present in patients with GBM, but they never metastasize. They never, virtually never, spread to other organs. So it's uniquely a totally isolated tumor. And the vast majority of isolated tumors can be cured now. So it really stands out as a remarkably different tumor with really different biology, difficult to study. If you ask the people that treat them, why do they recur? Because they always do. There's sort of two schools of thought. One is that so typically what they do is they'll do a surgical resection, then they do radiation, chemotherapy, and it comes back. And so one thought is that these are resistant cells. They just evolve resistance. The other thought is that they're actually the distant cells. Michalina talked about this, that they can roam far and wide in the brain, and that even when you remove the primary tumor, these guys kind of come back and magically appear in their homeland and recur. You would think after all these years, someone would actually have a definitive answer to that question, but there still is significant debate, as I understand it.
Fascinating. Thanks so much, Bob. Other questions or comments from people in attendance? A question I have, being very naive about these cancers in general, are glioblastomas seen in other non-human animals? And if so, do they show the same heterogeneity and same kind of weird characteristics where they don't metastasize but pop up in other parts of the brain and then come back? And yeah, just what's known about them outside of humans?
Yes, so they are actually they are well, let's say not common, but they do happen in dogs. So there is a really nice study by Roel Verhaak showing that actually the genetics of the canine GBMs are fairly similar to the human. So that definitely would be an interesting model to look at, because they arise spontaneously and they are also diverse. Because I think one of the limitations of our studies of GBM biology is that we are really still not having very good models for in terms of animal models to study the disease, because most of the murine models or the xenograft models are actually not disseminating into the brain parenchyma as you would see in the human tumors. So those are definitely not great to actually model this aspect of the tumor that may be very strongly contributing to the resistance to therapy. So yes, definitely the canine tumors are of large interest to the community to actually dig deeper into the diversity of those to understand really what's going on.
Great, thanks so much. Next we'll go to Michael. You can go ahead.
Yep. It's another question. I suppose the answer to this will be no, but is there anything known about biomarkers that are effective in detecting these tumors early? And if not, why not? Does it have something to do with the brain-blood barrier, or?
Yes, so I'm not aware of any sort of easy biomarkers that could be used to detect an early disease in case of brain tumors. And as Bob mentioned, that there is definitely circulating tumor cells that are disseminating from these tumors, and they are actually sort of really nicely found in the cerebrospinal fluid, but also in the blood of the patient. So the cerebrospinal fluid actually has a little bit of a better representation of the diversity of the actual tumor compared to the CTCs from the blood. So yes, it's possible that it may be just not disseminating into the blood to the same extent as the tumors that are housed in other organs due to the blood-brain barrier. That's possible. But in general, I think yes, early detection is sort of easier to build biomarkers when you actually have another method to detect those tumors early. And here sort of, I don't know whether there are any studies with incidental findings of sort of small or early developing gliomas. Usually they are like advanced from the time that we see them. So that's the challenge.
Great, thank you. I'm going to quickly go to one question in the chat, and looks like we're having some Zoom bombing issues. So I'll remove that person in just a moment. Sorry about that. But the question from the chat from Mary asking, does anything characterize the rare patients who do survive past 5 years?
I actually don't know. So there's definitely there are definitely some patients who do survive longer, but I'm not aware of any larger study that would actually look into the characteristics of these patients together. Because I think that's also one of the challenges, that in order to construct a really good clinical trial, to measure these things, you actually have to have sort of enough of the population, right? So you have to have the right numbers. And I think in the past, it has been a little bit challenging to construct those within the bounds of a single center. But I think right now there is a large push towards actually constructing those really large consortia that are looking across different centers with huge collaboration from all the large neurosurgical departments. So I think that's something that we probably are going to see in the future, but I'm not aware of anything known at this point.
If you don't mind, I can just throw in something that we've done some work on this. The MRI is the standard imaging tool. It's really the only way to evaluate the tumor over time. And we have looked at the characteristics of glioblastomas at the time of presentation to see if there's anything that predicted people that lived long. Unfortunately, the survival is no more than 2 years typically. So we looked at people who live 5 years. You can take, and Michalina, this may be a project that we could talk about, but you can take, MRI typically uses 12 different sequences. So you basically interrogate the tumor over and over again using different sequences, each one of which is slightly different in terms of what it's sensitive to. So you can combine these images to basically form a sort of every voxel has a kind of a vector that is related to the its intensity on these different sequences, and we've called these habitats. So you can look at heterogeneity in habitats, and it happens that one habitat was extremely favorable. If you had a fair amount of this, it was these people lived quite a long time. We think, but don't know for sure. It was a habitat where there was a lot of cellularity, but there was also a lot of edema and pretty good vascularity. We think that most likely that represented an immune response, and which would make sense. I mean, pathologic studies have also shown that the greater the immune infiltration, the greater the longevity of these patients. So, but that is something that we can now do MR guided biopsies and look at specific habitats and characterize their the cellularity of the tumor in that location, and perhaps begin to build on that to develop this sort of microscopic imaging and at multiple spatial levels.
Yeah, so that's definitely something that we're super interested in, and we're trying to do that on a small scale as well, trying to really understand what's, because I guess one issue with the sort of prediction of how the patient is actually doing based on the MRI, one of the challenges with that is that it's very difficult to differentiate sort of the pseudo progression from the actual progression, right? So then the first scans that the patient is getting after treatments are, it's really hard to say whether the patient is actually getting worse or better. So I think, yeah, that's definitely super interesting to actually get more.
Yeah, that's why we use pre-treatment only studies, but I agree that if you could start to look at how it varies over time. If you could map, so landscape ecology has developed metrics that allow you to take large spatial scale images like satellite images and build species maps, you know, within that by these and scaling those spaces, those spatial dimensions. And so we've been trying to do that, and we think that I think there may be a way then to be able to sort to characterize spatial heterogeneity at a molecular level if we can use that kind of technology and apply it to the large scale imaging, you know, like MRI.
Yeah, no, I think that will be great.
Great, thanks so much. Looks like we have another question from William. William, you can go ahead.
Thank you. Yeah, it's just a question. I've been at Duke for 30 years, so I have to ask about the immunotherapy with the poliovirus that they are developing here. So it's you infect the tumor, and in theory, you could get samples, and sadly at the time of autopsy, to look at sort of escape variants and things of that nature. So my question is, has your group or someone in the field kind of looking at the escape variants from the immunotherapy back to the evolutionary perspective? Is the immunotherapy causing evolution of the tumor? Has anybody looked at that? And if not, is somebody planning on doing that?
Yeah, so if I remember correctly, there's definitely at least one study that was looking at that, where they were looking at specifically which mutations were actually present in the tumors before and after immunotherapy, and basically trying to see whether there's any particular selection for certain genetic alterations. And I believe that they did see that PTEN was one of the genes associated with worse prognosis in terms of immunotherapy, but whether there were additional studies on that I'm not sure. I would definitely love to get my hands on samples from those immunotherapy trials to actually be able to look at just even the simple EGFR CDK4 and how that actually relates to to the outcomes, right? So I think, yeah, those trials definitely are going to be, I mean, that's exactly what my lab is trying to do, right? We're very heavily focusing on using FFPE samples, because those are the ones that actually are always collected at every single trial. So then if we can get, samples from these trials, then we'll be able to get really a lot of information about what may be driving the progression.
Great, thank you so much. So we do have a couple minutes left if there are any final questions, and my apologies about having to disable the chat, but do feel free to raise your hand if you have any final questions before we wrap up for today. All right. Well, not seeing any additional questions, I want to thank you again, Michalina, for a wonderful presentation and talk. This is the point where I typically share the Club EvMed website to show what we have upcoming, but our website is down for planned maintenance at the moment. So instead, I'll share our flyer. So this is the great talk we just heard. Our next Club EvMed will be with Henrik Schulenberg and Aditi Batra on the 24th. And that's going to be on sequential antibiotic therapy as a potent strategy against bacterial pathogens. That should be an excellent talk. And we do have additional talks lined up for April as well, which you'll be able to see on the website once it is back up and running here shortly. So with that, I want to thank you all again for attending, and I will send out a link to the recording soon. And we hope to see you in a couple weeks at the next Club EvMed. Thanks, everyone. It's great to see you.
Awesome. Thank you so much. It was a great discussion. Thank you. Bye.